Good day, everyone. This video will talk about the structure and the properties of nucleic acids and also the biological significance of nucleic acid in organisms. I'm Mr. Ong from College Tingkatan 6 Seliputra. Previous lessons, we have talked about the amino acid and also the proteins. So these videos will talk about the nucleic acid and the types of nucleic acid. Nucleic acids transmit hereditary information and determines what proteins a cell synthesizes. You are familiar with this diagram in which you can see in the nucleus, the DNA, are found in the nucleus, and then the mRNA is produced or is synthesized from the DNA, in which the genetic information in DNA is copied into the mRNA. Then the mRNA diffuses out of the nucleus through the nuclear pore to the cytoplasm and attached to the ribosome. In ribosomes, the tRNA carry a particular amino acid and binds to the mRNA in ribosomes to synthesize the polypeptides. The process to synthesize mRNA or RNA, from DNA is called transcription, in which the process of synthesized proteins is called translation. You will study these transcription and translations in semester 3 for detail. Today, we're just going to talk about DNA and RNA. Two classes of nucleic acids are found in cells. Deoxyribonucleic acid, or short form is called DNA, and ribonucleic acid, the short form is called RNA. So from this diagram, you can see the DNA actually consists of two polynucleotide strands that coil in an alpha helix form, in which the RNA also shown an helix forms, but with a single strand of polynucleotide. And from here, you also can see the DNA is longer than RNA. And these are the nitrogenous bases they found in DNA: cytosines, guanines, adenines, and thymines, in which the nitrogenous base found in RNA are cytosines, guanine, adenine, which are the same as in DNA, but the difference is the thymine is replaced with uracil. DNA composes genes. As you see, the DNA actually they're associated with protein histone, the histone protein, so to form the chromosome. If we remove the histone protein, so from here you can see the DNA, and then DNA composes genes. And then the genes contains information, genetic information, or the instruction for making all the proteins as well as RNA the organisms needs. So RNA participates in protein synthesis, in which the amino acids are linked, and then they're carried by the tRNA, and then they're linked to form the polypeptides. So this one is the primary structure of a polypeptide, the sequence of amino acids in the linear chain. They're joined by peptide bonds. Nucleic acids are macromolecules, same like the carbohydrate. They are polymers. So when they are polymers, that means they contain or they are made up by monomers. So the monomer for nucleic acids is known as nucleotide. So the many nucleotides that join together to form polynucleotide. So this is the structure of a nucleotide, in which they contain a pentose sugar, the pentose. So pentose means that you have learned in the carbohydrate subtopic. So for the nucleic acid, the pentose that involve the either is deoxyribose or ribose. Okay, depends on it is DNA or RNA. Then the pentose will bind to the nitrogenous base. Okay, with a nitrogenous base at the carbon 1, okay, at the carbon 1, and also with a phosphate group, okay, at the carbon 5. So this is nucleotide. Okay, this is a structure of a nucleotide. The combination of a pentose, okay, with a nitrogenous base form a compound called nucleoside. If we just look at the pentose, okay, the pentose that join or they combines with a nitrogenous base, so it is known as nucleoside. If the nucleoside bind with a phosphate group or 3 phosphate group, we call it as a nucleotide. Okay, so the specific name for a phosphate one is called a nucleotide monophosphate lah. So the nucleotide here is depends on the name of the nitrogenous base. Okay. So a nucleotide has 3 components, okay, as we discussed before. So the pentose is either deoxyribose, the one if in DNA. So it is a ribose if it is in an RNA. Then a nitrogenous base. So this nitrogenous base, like just now we have discussed, there are 4 types of the nitrogenous bases, okay, in DNA and in RNA. So it depends on which nitrogenous base binds to the pentose, okay, or to the deoxyribose or ribose. And one or more phosphate group which make the molecules acidic. So we know the phosphate group is acidic and with a negatively charged, okay, with a negatively charged, okay. So that means the nucleic acid are negatively charged. Okay, DNA negatively charged and ribose also negatively charged. The nitrogenous base may be either a double ring, we call it as a purine, or a single ring, we call it as a pyrimidine. So with 2 carbon rings, okay, 2 attached carbon rings, we call it purine, for example, adenine and guanine. Okay, adenine and guanine. Whereas the pyrimidines, the example of pyrimidines are cytosine, thymine, and uracil with a single carbon ring. DNA, okay, a DNA commonly contains the purine, which is adenine and guanine, and the pyrimidines, cytosine, and thymines, deoxyribose and phosphate. Whereas the RNA contains the purines, adenine, guanine, pyrimidine, cytosine, and uracil. Okay, so the thymines is instead with uracil. So together with ribose and phosphate. So the name for the nucleotides in DNA and RNA different. Even though they have similar, some of the nitrogenous base are the same. Okay, so for example, the DNA nucleotide, if the adenines they bounce to the pentose or deoxyribose, so we call this nucleotide as deoxyadenosine triphosphate. And this one is deoxyguanosine. Okay, this one is guanine. Okay, guanosine triphosphate. And if this is the thymine, so deoxythymidine triphosphate. And if this is the cytosine, is deoxycytidine triphosphate. And the short form, they have a small letter D in front. DATP, DGDP, and so on. So the D is stand for deoxy. In contrast, the nucleotides of RNA, okay, we also call it as the similar names, okay, as a DNA. The difference is there is no deoxy. Okay, there is no deoxy. So for example, adenosine triphosphate, guanosine triphosphate, and so on. So the short form is ATP, GTP, UTP, and CTP. So now you see the ATP. So you're quite familiar. I think you're quite familiar with this, with this name, okay? Because ATP, okay, ATP molecules are the energy currency, okay, or the energy, the storage energy molecules that synthesize in mitochondria during aerobic respiration. Okay, so that means this ATP that provide can supply energy for cell activities. So that one is actually they are that is what we call the one of the RNA nucleotide. Okay, the nucleic acids are made of a linear chain of nucleotides. Okay, as we see just now we say they are polymer. Okay, so it's the polymers called the polynucleotides. So this polynucleotide actually they contain or they are made up of what we call the nucleotides. Okay, they are made up of nucleotide. So the nucleotides are joined by phosphodiester bonds. Okay, phosphodiester bond. Okay, phosphodiester bond. So what is a phosphodiester bond? First of all, you need to identify a nucleotide. Okay, you need to identify the nucleotides first. Okay, so this here is one nucleotide. This is one nucleotide. Okay, then from one nucleotide, you can see the phosphodiester bonds that forms between the pentose, okay, between the pentose and the phosphate group. So they join at the 3' end here. Okay, so they form the phosphodiester bonds, okay, referring to these two. Okay, the condensation, okay, the formation of phosphodiester bond actually is through this process, condensation, okay, or we call the polymerization, okay, polymerization. So the condensation of nucleotides is repeated. So there is one nucleotide, so they join, they form the link with adjacent nucleotide with phosphodiester bonds. So this process is repeated, so to form a long, what we call a polynucleotide. Okay, so in this case, like here I show you, this one is ribose, okay, this is ribose, okay, because the oxygen is here. So when they form, actually in DNA also the same. So the 3' ends, okay, this is carbon 3, okay, carbon 3. Okay, the 3' end here, they join with the phosphate group, so to form a phosphodiester, referring to here. So does that mean this polynucleotide, this end is called 5' end. And then at the end here is 3' end. Okay, so in the condensation process, so that means they release or they produce one water molecules. And they remove two phosphate groups here. Okay, so since this one, the phosphate group, they link or they have the covalent bonds, so they contain energy. So once these bonds is cleaved, okay, or hydrolyzed, so they will release energy. So that means this reaction is an exergonic reaction, and the energy released is used to form phosphodiester bonds. Okay, so therefore the polymerization or the formation of polynucleotide, okay, in either RNA or DNA, they do not require additional energy from outside, okay, from an external source, because the process itself is an exergonic reactions. Okay, so as you see, the adders to the 3' ends here. Okay, and these strands must be paired with the DNA template strand. So that means here they have another DNA template strand. If they're DNA, so you can see later the nitrogenous base, they must bear a complementary base pair with the nitrogenous base in the template strand, okay, or the complementary strand. So like here, this one is for the DNA. Okay, for DNA, So this is the template strand. So this is the new strand they form. So from here you can see the new nucleotide there join to the adjacent nucleotide. Okay, so to form a polynucleotide. So at the what we call the 3' ends. So here is a carbon 1. Okay, here is a carbon 3 and carbon 3 this way. Okay, so here is a carbon 1. Here's a carbon 3. Okay, so from here you see, have you see this template strand and the new strand or the new complementary strand that form? They are in the opposite directions. Okay, the new strand they form, they must in the 5' to the 3' direction. Okay, in the 5' to 3' directions. And then from here, have you realized that the guanine, nitrogenous base, they always complementary base pair with cytosine. See? Yeah. And the thymine always complementary base pairs with adenine. So this is why we need a template strand, okay, to synthesize a new polynucleotide or the new complementary strand. So here is 5', okay, to 3'. So in opposite direction. Why like this? Okay, later will be discussed. So from here, as you see, the 2 phosphate group are removed. Okay, so this process is an exergonic reactions. So they do not need an additional energy because the release of the 2 phosphate group here, the pyrophosphate, they will provide or supply energy, okay, to form the phosphodiester bond. So that means the phosphodiester bond actually is formed, okay, to join the 2 adjacent nucleotides together, okay, at the between the phosphate group with the 3' end of the pentose. So if the one is DNA, so it's with a deoxyribose. If there is a RNA, so with 3' end of the ribose. Okay, then from here you see this is DNA single strand, and then from here you can see there is this one is called the phosphate sugar backbone, okay, because they have the same, and then the difference is on the nitrogenous base are different. Okay, same to the RNA, same to the RNA. This one is the sugar, the phosphate sugar backbone. Okay, so either DNA or RNA, they are unique, okay, defined, okay, by each specific sequence of nucleotide. And the sequence of this nitrogenous base, so the unique sequence of this nitrogenous base, so actually they constitute a kind of code, okay? And then this sequence of the nitrogenous bases, so they will determine the primary structure of a protein or polypeptide, okay? That means the amino acid sequence in a polypeptide. Okay, in overview, let us discuss, okay, about the differences and also one of the similarity of the DNA and RNA. DNA, the pentose is deoxyribose, RNA is ribose, and then the nitrogenous bases that found in the DNA is a CGAT, and the nitrogenous base they found in the RNA CGAU. So the difference is the thymine and the uracil. And the DNA has a double strand. Okay. RNA has only a single strand. DNA has is a long chain of the long chains of polynucleotide. RNA is a shorter chain, the shorter chain of the polynucleotide. DNA is synthesized in nucleus, okay, and a nucleoli, okay, the ones for the prokaryote, and also in the chloroplast and in the mitochondria. Okay, same to the RNA, they also synthesized. They are synthesized in nucleus. Okay, not nucleus. This is in nucleus. Okay, so the nucleoid and this one, reactivity, DNA normally is less reactive compared to RNA. Okay, but the DNA is more stable in alkaline condition, okay, compared to the RNA. But the DNA can be damaged easily by UV ray. But the RNA is more resistant, okay, to the damage by the UV rays. Okay. And function, DNA, they carry the or contain hereditary materials. And the RNA actually is used for protein synthesis. DNA, okay, the replications of DNA, that means the synthesis of DNA occurs during cell division, okay, by mitosis. Whereas in the ribonucleic acid, okay, in the ribonucleic acid, okay, this one meiosis also got, okay, they want to produce, okay, they want to produce what we call the additional DNA strands. And in RNA, the RNAs are continuously synthesized, okay, and break down and reuse, okay, when they want to synthesize the proteins, okay, or they are very active in protein synthesis or hormone synthesis. Okay, RNA is composed of one polynucleotide only, okay, in the helix form, whilst the DNA consists of two polynucleotide chains. They also forming a double helix, okay. And this DNA, because they contains two polynucleotide chains, so the hydrogen bonds that are formed, okay, between the nitrogenous base, okay, between the nitrogenous bases of the two strands, okay. And the DNA structure that we learned so far is based on the Watson and Crick model. Okay, James Watson and Francis Crick, working at Cambridge University in 1953, they proposed, okay, the structure of the double helix of the DNA. Okay, the DNA structure with a double helix. Okay, so this is the models, okay, that they are proposed, and then the time they won the Nobel Prize. And actually, Watson and Crick, they just combines the work, okay, or the discovery from Rosalind Franklin, okay, and the Erwin Chargaff, okay, to deduce the structure of the DNA model, okay, their DNA model, okay. So Rosalind Franklin's work in 1953. So she used X-ray diffractions to reveal that actually DNA, they have the regular shape, okay, or the regular structure, which is like the helix form, okay, like a helix form. And in 1947, American scientist named Erwin Chargaff, okay, discovered that the amount of guanine, okay, the amount of guanine and the cytosines, okay, nitrogenous bases are equal in any sample of DNA. So from here, we can come up, that means A also, the adenine also have the same or the equal number with the thymines, right? Okay. So based on these discoveries, Watson and Crick propose or they built, okay, the DNA structure model based on these discoveries. And then here you can see there is a sugar phosphate backbones, okay, sugar phosphate backbone on the outside of the helix, outside of the helix, okay. And the nitrogenous bases are paired, okay, they are base pair, complementary base pair in the interior part, okay, interior region of the DNA molecule. Okay, the two sugar phosphate backbones run in opposite directions. Like here, you can see here is a 5'. They run in the opposite to 3' here, 3' end. And then the other strand, they run here 5' end to the 3' end here. So they run in parallel, okay, they parallel, but in opposite direction. Therefore, we refer this situation, okay, or this arrangement of the polynucleotide in DNA as anti-parallel. Okay, anti-parallel. Therefore, each end of the double helix must have an exposed 5' end phosphate on one strand and an exposed 3' end on the other strand here. Okay, so here 5', here 3'. Why we call it as a 5' end? Because here is the carbon 5. Okay, why we call here is a 3' end? Because the point here, the last one, is the 3' end. Okay. Only certain nitrogenous bases in double helix are compatible with each other. This was according to the Chargaff rules. So the adenine, okay, adenines are always complementary base pairs. Okay, in exam we use the terms complementary base pair with thymines, whereas the guanines always complementary base pair with cytosine. So therefore, the the number of guanines is always equals to the number of cytosines in the DNA sample, and also for the adenine and thymine. The purine, okay, adenine and guanine base pairs with a pyrimidine, okay, the cytosines and the thymines, again, by hydrogen bonds. Okay, so for the guanine, you see the guanine, they base pair with the cytosine with 3 hydrogen bonds, whereas the adenine, they base pair with the thymines with only 2 hydrogen bonds. If we were to read the sequence of the bases along one strand as we travel okay, from the 5' end to the 3' end, so we will know, okay, we would know the sequence of the nitrogenous bases on the complementary base pair, on the complementary strand, okay, on the complementary strand. Okay, for example, here if here 5' end to the 3' end is a A, C, T, G. So from the other strand, we would know the basis sequence. The basis sequence. So here A always base pair with T, and then C with G, T with A, and so on. Okay, so you can see this is like this. So this one have 2 hydrogen bonds, this one 3 hydrogen bonds, 2 hydrogen bonds, 3 hydrogen bonds, 3 hydrogen bonds, 3 hydrogen bonds, and so on. Okay, so the DNA double strand, they are anti-parallel. Okay, and the nitrogenous bases are always complementary to each other. Okay, so the 2 polynucleotide strands of the double helix, okay, are complementary, each the predictable counterpart of the other. If one strand has the base sequence, okay, like this one, okay, so you must know whether it's the 5', where is the 3' end. Okay, then the complementary base pairing, okay, will inform us the anti-parallel strand, okay, the anti-parallel complementary strand will be like this. So again, determines the 5' and the 3' end. So they are anti-parallel. Okay, anti-parallel. In other words, in double-stranded DNA molecules, the number of purine equals, okay, they're equals to the number of pyrimidines. So that means the A will equals to the thymines, and the guanine also equals to cytosine. So this equalities is known as Chargaff rule. Okay, this table show the ratio between the A and T, G and C. So from here you see, even though they're slightly different, but they were not far from 1.0. So from here we can say they are not significant difference. So we accept that the A is equals to T and G is equals to C. So the difference here is just due to because sometimes they have extra different strength, the nitrogenous bases. Okay, these features enables DNAs to copy each gene precisely for inheritance. Okay, these are what we call the Chargaff rules. So in preparation for subdivision, each of these two strands of a DNA molecules, they serve as a template. Okay, like just how we see. So one strand was serve as a template, okay, for the new complementary strands to form. Okay, so the blue color one is the template. Okay, so here is the template from the parental DNA. Okay, and then each of these strands also later will becomes the template for their daughter DNA they form. Okay, so therefore the DNA they form, okay, the double strand DNA, and they form, they are identical. Okay, they are identical. Okay, one long DNA, double helix DNA includes many genes. Okay, one DNA they may contain many genes. Okay, so the two polynucleotides are held together, okay, by the hydrogen bonds here. Okay, so inside your nitrogenous base, you can see the nitrogenous base. Okay, they're held together by the hydrogen bonds. Okay, don't forget here's two hydrogen bonds, here's three hydrogen bonds, and also by the van der Waals interactions. Okay, because most of the interior region of the DNA molecules, okay, contain non-polar molecule, so they will have the van der Waals interaction when they comes close contact. And the hydrogen bonds between the complementary bases is weak. Okay, it's weak and can be easily broken down, okay, by heat, okay, into two strands. However, the phosphodiester bonds are strong covalent bonds, and then they can stabilize the DNA molecules. And also, if the DNA is inside in the nucleus, okay, or in the cytoplasm, so because the nucleus and nuclear plasm and the cytoplasm, they contains aqueous solution. Okay, they are in the aqueous environment. So the surrounding water will exclude the DNA molecule. Okay, they will exclude the DNA molecule. So so that means they will have the hydrophobic interactions. Okay, so we already learned the hydrophobic interaction in the previous lesson in the subtopic of proteins. So due to the hydrophobic interactions, so the sugar phosphate backbone, so the phosphate part, they were at the outside here. So that means they're contact with the water molecule. And then the interior ones are the most of the non-polar molecule. Okay, so therefore, when they have the hydrophobic interaction, and then they'll make the non-polar molecules closed contact, and therefore will generate these van der Waals interactions. Okay. All right. DNA is transcribed to form RNA. We have learned the DNA structure. Now let us discuss about the RNA structure. RNA actually is transcribed from DNA. And then there are three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. So this is the structure of mRNA. This one is the structure for the tRNA, and this is the structure for rRNA. Okay, messenger RNA, or we call mRNA, is a single strand of RNA, okay, that carries hereditary information from gene for synthesis of proteins. Then the mRNA will attach to the ribosome, okay, for protein synthesis. So from here you can see this is a double strand of DNA. Okay, so like here in order to produce an mRNA, we want to see which strands will be used as the template and which strands will contain the information, the genetic information to be transcribed and translated. So for example, in this diagram, they show that if the cells they want to translate this strand, the upper strand, okay, so this strand is called a coding strand. Okay, if we want to translate this strand, the information, genetic information from this strand, we must use the complementary strand as the template strand. Okay, so that means when we use this as a template strand, so we can synthesize an mRNA which contain the genetic information same like the coding strand. You look at the information, they are the same. A, C, T, T, they instead with U because you reserve in mRNA or in RNA, there is no timings. So G, G, C, 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 and so on. Okay, so this is the template and the coding strings. Okay, so when the mRNA is synthesized, okay, once the mRNA is synthesized, then we can see the sequence of 3 consecutive bases in mRNA, okay, in the mRNA are known as codon. Okay, all of these are triplet base are called codon. And then each codon was specified for 1 amino acid, okay, except the stop codon. So for example, one codon that corresponds to the amino acid leucine is CUU. Let's say the one is a CUU, so we will know this codon is code for amino acid leucine. Okay, so how we know which codon is for which amino acid? Okay, so we can look at this genetic table. Okay, this is known as the genetic code. Okay, the table of genetic code. So like here, first base, second base here, and then third base here. Okay, like just now we've mentioned CUU. Where is CUU? C, the first base is C, second base is U, then the third base also U, so CUU. Okay, so it's leucine. So this is the codon on the mRNA. Okay, so this codon is something like the words of genetic information. Okay, so each nitrogenous base is like the alphabet. Okay, so that means in the genetic information, okay, all the words they have only 3 alphabets. They consist of or made up of 3 alphabets. Okay, no 1, no 2, no 4, no 5, and so on. Okay, so when all of these words they join together, so this will determine the primary structure of a functional protein, which is the amino acid sequence in a polypeptide. Okay, so you will study this genetic code in detail in the semester 3 Okay, here I just show you how to relate the codon, okay, with the genetic code and the amino acid, and then later with the tRNA and the anticodon. Okay, because each codon, okay, consists of 3 nucleotides, so the code is described as a triplet code. Okay, sometimes we also call the code as a triplet code. Okay, and this code is universal. Universal means the same code encodes for the same amino acids in almost all organisms. Okay, not all, it's almost only. Okay, so for example, plants and others, animals, they also the codon, let's say like just some CUU code for leucine. So in other animals, CUU also code for leucine. In plants, they also code for leucine. In bacteria, they also code for leucine. Same. So that's why we call this genetic code is universal. Okay, since we already go through what is mRNA and the structure, the mRNA contains a codon, and then what is the function? What is the role they play by the codons? How to relate to the DNA and the genetic information in DNA. So now we look at the transfer RNA. Okay, the short form is called tRNA. It is a single strand of RNA. Okay, but it can fold back on itself. Like here also 5' to 3' end. Okay, only they can fold back. When they fold back, and then they are helped by hydrogen bonds. When you see united hydrogen bonds, so they have to form a specific shape, and then this one is called tRNA. Okay, so this one also tRNA, but in the 3-dimensional structure lah. Okay, from here you see, first of all, you must know where is the 5' end and where is the 3' end. So 3' end at the higher than the 5' end here for the structure. And here you can see there is one triple code, we call it as an anticodon. What is the relationship between anticodon and codon? Okay. The anticodon, okay, here is the anticodon. They were complementary base pair with the codon on the mRNA. You see, the mRNA here is CUU, so that means the anticodon on the tRNA is GAA. So that means the transfer RNAs are crucial part of decoding machinery because they act as adapters. Okay, because they want to translate what? They want to translate the genetic information on the mRNA, okay, to synthesize a polypeptide. So this is anticodon is something like the adapters. So a particular tRNA, they can recognize a particular codon, okay, because it has a sequence of 3 bases known as anticodon, which is complementary base pair with the mRNA. All right. Then each tRNA, they can link with a specific amino acid to form a tRNA amino acid complex and recognize the appropriate codon for the particular amino acid. Let's say this tRNA, okay, they carry a leucine, okay, with an anticodon GAA. So this tRNA, they only can complementary base pair with the codon CUU. CUU. Because the codon is CUU, so we will know, okay, this tRNA, they will carry leucine, okay. Because here's GAA, so the amino acids, this genetic code is for mRNA, okay. So from this here, we also can determine the anticodons, okay. If an anticodon, okay, here let's say proline, proline, this codon, codon proline CCG. So that means the anticodon will be GGC. So that means the tRNA, okay, here is a 5' end. Here's 3' end. So here anticodon is antiparallel also. Here 5', here 3'. So that means the anticodon with this one CGG or GGC here easier for us to know which amino acid and the codon. Okay. So the tRNA with GGC, okay, or CGG here, they will carry proline amino acid. Okay. So from here we know from here we will know which amino acids will be carried by the tRNA. Okay. So for example, GAA or AAG here, then from here this anticodon, so from here we can come up with the codon. Okay, once you have determined the codon, so and then we can refer to the genetic code table, okay, and then to find what is the amino acid that carried by that tRNA. Okay, so this degeneracy or the redundancy. This one will be discussed in the semester 3. So therefore, there are more kinds of tRNA. Okay, this one also the redundancy one. All right, now we already know the function of the tRNA is to carry the particular amino acid that regarding to the codon of the mRNA. Then how about the function of ribosomal RNA? Ribosomal RNA, also known as rRNA, is produced in a nucleus within the nucleus, and it is an important component of the structure of ribosome. Okay, a ribosome actually they consist or they're made up by two subunits. One is called large subunit, and the other one is called small ribosomal subunit. So each subunit they actually made up by RNA and protein. Okay, so the gray color one is RNA and the purple color protein, and this one gray color RNA, blue color is protein. So the small ribosomal subunit also made up by the RNA and protein. And then these two subunits, they join or they combine together will becomes a functional ribosome. Okay, a functional ribosome. So that means the rRNA actually they are the important components to make the ribosome. And then the rRNA, they have catalytic function. So that means this rRNA is an enzyme. They are the enzymes that used during protein synthesis. Okay, so for this chapter, you just need to know the function of ribosomal RNA. Okay, they are the one of the component in a ribosome, and also they have the catalytic function, okay, in protein synthesis. So the detail how you will study in semester 3. Therefore, the ribosome is the site of the translation. The protein synthesis translation is the process of protein synthesis. So ribosomes are the organelles composed of two different subunits, like just as we've mentioned, okay, each containing protein and RNA. So the ribosome attached to the 5' end of the mRNA, okay, the mRNA 5' end, and travel along, okay, the ribosome will travel along the mRNA towards the 3' end. And then at the same time, the tRNA will carry the particular amino acid and bind to complementary base pair to the codon. Okay, and then to synthesize a polypeptide. Okay, so that means the sequence of the codon here, they will determine the sequence of the amino acid. Okay, so this is the first amino acid, this one the second, and so on. Okay, so this is a translation, okay, and overview for the process. So first of all, like the attach of the form, and then the new one, the new tRNA will carry the amino acid, so they'll join to the particular or an appropriate codon. Okay, this one is codon, not anticodon. This is wrong already. Okay, here this is anti. Okay, this one is anticodon. So this one is codon. Okay. All right. So this one will come to join. Okay. So then the polypeptide. Okay, they'll transfer to the new one. Okay. So here is the first one. This is the first, second, and so on. So and then after that, this tRNA will be removed. Okay. The acid the ribosome, and then after that, this one will come in, and then the new one will come in, and then they join. So this process is repeated. So for this chapter or for this semester, you no need to understand the process for this translation. Okay, I show you this picture is for you have a better understanding. Okay, when you want to study the structure and the biological significance, okay, the function and the role for the DNA, mRNA, tRNA, and rRNA. Okay, so by from this diagram, so we can see better the function for each of these what we call nucleic acid that play their main role. Okay, so the rRNA is found in the ribosome. Okay, so translation or the protein synthesis actually required an RNA. Okay, and mRNA they carry the hereditary information. So where is the hereditary information come from? From DNA. Okay, from DNA. All right. So DNA. And then tRNA, they'll carry a specified amino acid. So based on the anticodon, okay, based on the anticodon, okay, and then join to the appropriate codon on mRNA. And then the ribosome actually they're made up by the RNA. Okay, so from here it's very clear the function for each of these or the biological significance for this nucleic acid. Okay, so these are the checklists. Okay, whether you can describe the structure of a nucleotide or not, and then how's the phosphodiester bond, phosphodiester bonds are formed, okay, in a polynucleotide, and you are able to distinguish or differentiate between DNA and RNA, and also to distinguish the types of RNA, okay, mRNA, tRNA, and rRNA. And also you must able to describe the structure of DNA based on the Watson and Crick model. Okay, and this one, okay, this one come from here. All right. So I think that's all for this video. And for the next lesson, we'll talk about the analytical techniques that use in biological molecules. For example, paper chromatography and the electrophoresis they use to separate, okay, proteins, and also to separate amino acids. Okay, they're different. And also the electrophoresis they use to separate nucleic acid, especially DNA. So that's all for this video, and thank you very much. Bye.